All right. Welcome to part 2 of the chapter on biology of the mind. Let's begin. There we go. All right. So when we talk about brain biology, we have to start with some of the early ways that we learn things about the physical structure of the brain. So we can go back hundreds and thousands of years, all the way back to Hippocrates. And Hippocrates is a very famous name in the medical world. If you've ever heard of the Hippocratic Oath, that comes from Hippocrates, who in a lot of ways was kind of the grandfather of modern medicine. Hippocrates was a very curious person. He was very curious about the human body. And he was one of the first to notice and record that if you punctured a person's skull and that person was suffering from brain swelling, that hole in the skull would allow some fluid to leak out and it would solve the problem of brain swelling. So people who are suffering from this condition, they might have severe headaches, they might not even be conscious, but Hippocrates found that if he were to put a little hole and allow that fluid to drain out, that lo and behold, some symptoms would be cured. Obviously, you now have a hole in your head, which isn't the greatest thing, but hey, this is early prehistory. So Hippocrates is really the first to make this connection between mental symptoms, things like headaches and the inability to focus and concentrate, and the physical brain itself. So the idea that the mind is housed in the head is sort of like the first recorded instance of it. So fast forward several hundred years, a couple centuries, and we start to learn about the brain through lesions. And a lesion is just a really broad general term for brain injury. A lesion occurs because of a brain injury, and a brain injury can occur because of an accident, a traumatic blunt force trauma to the head, a stroke. A stroke occurs when blood vessels are clogged or blocked for some reason, and blood can no longer get to certain parts of the brain. And blood actually carries oxygen and nutrients to different parts of our body, including the brain. So if parts of the brain can't get blood, they can't get nutrients and they can't get oxygen, and they actually die off. And then there are certain diseases that attack brain tissue as well. Some very famous case studies in brain lesioning studies are listed on the slide there. First, we have Phineas Gage. And Phineas Gage is in the picture on the far right of the slide. Phineas Gage was a man, he was a railroad worker back in the 19th century. He was involved in a railroad accident involving a railroad spike, which he is holding in his hand, and some dynamite. And as a result of that accident, that railroad spike that he has in his hand was lodged through his left eye. You can see that his left eye is actually closed in that picture because he no longer has one. The railroad spike entered his left eye and went up through the top of his head, just behind his forehead. So when this happened, everyone thought he was dead. I mean, he was laying on the ground with a giant railroad spike literally through his skull. In a few minutes, he regained consciousness and was speaking to the people around him. He seemed to be vaguely aware of what had happened. Obviously, a little bit concussed, so not quite fully sorted out yet, but he was aware of his surroundings, and this was just amazing. People thought he was dead for sure. So after Phineas Gage had his accident, his behavior changed pretty dramatically. Before the accident, he was known for being a stand-up guy. He was a good worker, showed up on time every day, went to church, paid his taxes, all good things that citizens do. Well, after this accident, he started showing up to work late, if he showed up at all. He stopped going to church. He stopped paying his taxes, and he was actually put in jail for a couple minor crimes. He started cursing a lot. So just a totally different person. And after his death, an autopsy was performed, and people actually looked at what his brain looked like. And his prefrontal cortex is totally destroyed. So the prefrontal cortex is the most forward part of your brain. It's right behind your forehead. And that part of him was just completely destroyed. So this was the first piece of evidence, and some very good evidence, that the prefrontal cortex plays a pretty important role in our personality, in who we are. And we'll talk about this later, but it actually plays a really important role in impulse control, which is a good explanation for why Phineas Gage was just a totally different person, kind of a jerk who couldn't filter himself after that. Next up, we have Tan. Tan is the name of a patient who had some severe speech problems. We'll talk about Tan when we get to the language chapter in depth, but Tan is the famous patient of Dr. Paul Broca. We'll talk about Broca's aphasia later on. But Tan was an individual who came to Dr. Broca for help, and Tan could only say the one syllable Tan. That's literally all that he could produce with his articulators. Tan, tan, tan. He could understand speech just fine, but he had severe speech production issues. And the picture in the middle is a picture of Tan's brain. And if you look closely, you can see some pretty severe damage right here. That's definitely a very large lesion. So that's not what a brain is supposed to look like, especially right there. So once again, we have a specific area of the brain that's damaged, and we seem to have some specific difficulties in behavior that arise because of that damage. So we have pretty good evidence that this part of the brain is involved with speech production. And then the last person we have listed here is HM. HM passed away a couple decades ago, and now we know his real name, Henry Molaison. HM is one of the most famous patients in the entire history of psychology. HM, because of an early experimental surgery in his life, he had his hippocampus removed. And because of the removal and destruction of his hippocampus, he suffered some severe memory problems after that. He seemed to have mostly intact long-term memory, but his short-term memory was completely destroyed. So you could have a conversation with him, and he would have no memory of that conversation 10 seconds after it happened. Researchers who worked with him for decades, he had to be reintroduced to who they were every single time that they came into his place there. We'll talk about HM at length later on. But these are some of the famous case studies that were early examples of how different parts of the brain affect behavior. And this is how lesion case studies work. You damage a part of the brain, and then you kind of see what happens. Obviously, in humans, you try not to damage it intentionally. But in the case of HM, sometimes it's done just out of, I guess, ignorance. All right, other ways that we learn about the brain. Direct stimulation. This is a slightly barbaric way, but it's still used today. And if you remember from part 1 of the biology unit, this is how we learn about the motor cortex and the sensory cortex. So basically, in direct stimulation, the patient is awake, they are conscious. You pull back the skull and you electrically stimulate the surface of the cortex. And you do this to basically see what happens. So there's a very famous paper published decades ago by Roberts Bartholomew, who basically did this approach on a corpse, a fresh corpse. He pulled back the skull, had the motor cortex exposed, put electrodes into different parts of the motor cortex, sent electrical stimulation along those electrodes, and just saw what happened. So if you stimulate this part of the motor cortex, the left pinky finger twitches. Okay, write that down. If you stimulate this part of the motor cortex, the right elbow twitches. Okay, that's pretty good evidence that this part of the motor cortex controls whatever part of the body just twitched. like I said, pretty barbaric, but I would say less barbaric than something like a Phineas Gage situation. All right, moving on to more modern day methods. I had to get off that screen with that exposed brain right there. So, more modern methods. We have brain imaging techniques. And we're going to talk at length about these imaging techniques. And to start, I want to set up two broad categories for these techniques: structural categories and structural techniques and functional techniques. And we can further split up functional techniques into spatial resolution and temporal resolution. So this will make a little bit more sense later on when we actually talk about techniques. But structural techniques basically tell us how the brain looks, where healthy tissue is, where damaged tissue is. Functional techniques tell us how mental processes are occurring. And like I said, you can split up functional techniques into two different categories of spatial resolution and temporal resolution. Functional techniques that give you spatial resolution tell you where a process is occurring in the brain. So if you have a spatial resolution technique and you give someone a reading task and you put them in an imaging method, and lo and behold, a particular part of their brain lights up, it's a good bet that that part of the brain is involved with that reading task. Temporal resolution deals with when brain activity occurs. So temporal refers to when, spatial refers to where, and then structural refers to just healthy versus unhealthy tissue. Okay, let's start with structural. Two examples would be a CT scan or an MRI. I bet there are people in this class who have had one or both of these techniques before. A CT scan or an MRI, they can tell you things about the physical structure of the brain. They can also tell you things about the physical structure of other parts of your body. Maybe you've had an MRI done on a shoulder or a knee or maybe a hip or something like that. Medical professionals will use this information to see the extent of damage or whether or not damage has occurred. So a CT scan uses x-rays, and if you've ever seen an x-ray before, I am not a medical professional, so I can't really make sense of x-rays, but if you've seen one before, you know that at least professionals are able to look at one and tell you what it is healthy, what is not healthy in that picture. An MRI stands for magnetic resonance imaging, uses a much different technique, which we'll talk about for fMRI. All right, those are the only two that I want to cover for structural. They're not all that interesting from a psychologist point of view. We're more interested in functional techniques. So again, functional techniques tell us about brain activity, not just brain structure. And we can talk about temporal resolution, which is when brain activity occurs, or we can talk about spatial resolution, which is where brain activity occurs. So an example of some technique that gives us temporal resolution would be an EEG, electroencephalograph, and a couple examples of techniques that give us spatial resolution are an fMRI and a PET scan, positron emission tomography. All right, let's talk about these techniques specifically. So for structure, we have a CT scan and an MRI. For function, we have an EEG under the temporal resolution category and fMRI and PET scan under the spatial resolution category. Nice little summary slide there. So it's safe to say that psychologists are typically interested in both temporal resolution and spatial resolution. So in order to get that information, we usually combine the techniques. We'll combine an EEG with an fMRI or a PET scan. All right, let's move on. Let's talk about these techniques. All right, so an EEG stands for an electroencephalograph, and this is a picture of an EEG in action. You have the subject or the participant put on this cap. The cap is full of these electrodes. And these electrodes measure the electrical activity just under the skull on the surface of the cortex, wherever that electrode is placed. So this technique gives us a very high degree of temporal resolution. It can tell us very quickly and very accurately when brain activity is occurring. It can pick up on those little action potentials incredibly accurately. But it has very low spatial resolution. It can't tell us where that activity is occurring with a high degree of accuracy. And you can kind of see that looking at this cap. You can see all these electrodes, and let's just look right here, right between the eyes, right in the middle of the forehead. You've got 4 electrodes here. Those 4 electrodes are covering about half of the prefrontal cortex. So between 4 different location points, that activity could be occurring anywhere within a small radius around that electrode. When we talk about spatial resolution, we want to have a much higher degree of accuracy than this little cap full of electrodes can give us. So EEGs are measuring what we call event-related potentials. Now you remember from the last lecture, potential is just a fancy word for electrical charge. And event-related means that it is an electrical charge. Let's just swap out potential with electrical charge. It is an electrical recharge in response to an event. It's related to an event. So what you do in EEG experiments is you give the person a task, or let's say you show them a really provocative picture. Maybe it's a lewd picture or something like that. And you simply measure their reaction to it. They don't even have to respond out loud. They don't even have to say anything. You can measure their response to that task or that picture using this electrode hat. And you can measure the event-related potential in response to that specific picture or task. The EEG is functional. It tells us something about brain activity, and it is temporal. It has a high degree of temporal resolution. It tells us when brain activity is occurring. What about where? For that, we can use something called PET scan, positron emission tomography. Some of you may have had one of these. They are a little bit less common than an MRI or a CT scan. A PET scan occurs when a radioactive agent is introduced to the body. That agent is then metabolized where brain structures are active. So you're basically injected with a radioactive dye, and that radioactive dye makes its way through your bloodstream. It crosses the blood-brain barrier into your central nervous system, makes its way into your brain, and then you are given a task. And while you're doing that task, your brain is being scanned for where concentration of that radioactive agent is. And the idea here is that parts of your brain that are using a large amount of blood will have a large amount of this radioactive agent in them, which means that those parts of your brain are necessary to complete whatever task it is that you're given. So on the right-hand side of the slide, this is actually a PET scan image. And the dark red areas are areas where that radioactive substance is in high concentration, and the blue areas are areas where the radioactive substance is in lower levels of concentration. So this task looks like it's using quite a few different areas of the brain. Back here towards the back of the brain, this is where the visual cortex is housed, so I would guess this is some sort of visual task. We've also got a pretty dark area over here. That would indicate to me that it's probably the motor cortex, and since control is contralateral, this would be the left side, so they're controlling the right side of their body. Usually people are right-handed, so this might be a writing task, or maybe they're using a mouse on a computer. Really no way to know, but you can see that there's differing levels of concentration of this radioactive substance. So to summarize, PET scan, it's functional, it tells us something about brain activity, and it's spatial. It tells us where that brain activity is occurring. All right. This slide is a little bit out of order. MRI, so it stands for magnetic resonance imaging. And here's a picture of an MRI. This is actually an MRI of one of my previous advisors' brain. This shows the contrast between soft tissues and structures. And this is actually an image of a pretty healthy brain. You can see the cortex here is sitting on top of the limbic system, and you can see the brain stem is below that, and you've got the lovely little cerebellum here, and you've got someone's nose, mouth, articulators, throat, all that good stuff, safely encased inside this bony structure that we call a cranium. Again, an MRI just tells us structural information. Very useful, but not all that useful for most psychologist needs. So an fMRI stands for a functional magnetic resonance imaging. Look at that fancy word that they stuck in there, functional. So it's an MRI, but it tells us something about the brain activity that's occurring in the brain. An fMRI is, it sort of was a rock star technique for a couple decades. It's kind of recently fell out of fashion, but it was revolutionary when it came out. It could give us structural information and functional information at the same time. It's an MRI, so remember, it's got that structural technique or structural component. The functional side of it here is more spatial than temporal. It can tell us where that brain activity is occurring, but it tells us where that brain activity is occurring with about a 4-second lag. So the brain activity occurs, and then 4 seconds passes before it is registered with the fMRI device. That might not seem like a long time, but when it comes to psychological processes, 4 seconds is pretty much an eternity. Our brains operate on such fast scales. Go back to last week's or last lecture, and we were talking about action potentials occurring over the course of 7 milliseconds. So you can have an action potential in 7 thousandths of a second. So you can have many action potentials over the course of 4,000 milliseconds, which is 4 seconds. That being said, still a very useful technique. This is an image of an fMRI. This would be the front of someone's head down here. Back here we have the back of their skull. And again, this would be the occipital lobe where the visual cortex is housed, and the orange areas are looking like a lot of activation back there. Pretty safe to say this is a visual task. All right, let's talk about some very new techniques that are kind of on the cutting edge of this part of psychology. Transcranial direct stimulation. It's also sometimes called transcranial magnetic stimulation, TMS or TCDS. And the basics of this is that you have a little wand, like a handheld wand. If you've ever seen those virtual reality setups where you have a visor that you put on and then you've got two kind of goofy looking video game controllers in your hands, that's kind of what this wand looks like. So you put the wand over a specific part of someone's skull and you activate it. And when it's activated, it sends very rapid, low voltage electrical impulses from that wand through the person's cranium onto the surface of their cortex. And you usually do it for two different reasons, or you can have two different effects. Number one is that it stimulates inactive neurons. So neurons that are just resting, they're hanging out at that resting potential. If they're struck by these electrical impulses, they can actually have action potentials where they wouldn't be having them earlier. So you can do this and you can kind of be like a mad scientist with this. You can cause someone to have a twitchy arm, or if you watch the video that will be linked on the Canvas page, you can see that you can disrupt the normal functioning of a task like they do in the video down here. So in this task, they have someone speak or sing. and they stimulate the same part of the cortex during both of those tasks. And you see that the person has immense difficulty being stimulated in that part of their brain while they are speaking, but they don't have any difficulties at all singing. So what this says is that singing and speaking occur in two different parts of the brain. But if you would like to watch that video, that one will be linked as well. So that's TCDS, and again, it's kind of the cutting edge. We don't know a whole lot about really anything about it yet. I mean, obviously we know something about it, but it's kind of the wild, wild west right now. It's actually a little bit difficult to get like ethical permission from review boards to use TCDS because it's just one of those things we really just don't know much about. There's actually a growing YouTube community online of people who create these little devices at home through little DIY setups, and they use them on themselves. They're kind of like using their own brains as experimental participants. I certainly don't recommend it. All right, so those are some basic imaging techniques. We talked about structural techniques, we talked about functional techniques, spatial resolution, temporal resolution. And now I want to move on to brain plasticity. Brain plasticity is our ability to change. Brains are incredibly plastic little critters. They're very, very fluid, very, very flexible. However, this plasticity ability does decrease with age. So first of all, plasticity involves the ability to recover from injury. And I just have to talk about Phineas Gage again because this is such an incredible case study. So here he is in a different picture holding the railroad spike that again punctured his left eye and exited the top of his skull. And here on the right side of this image, we have some, they're not actual MRIs or not actual CT scans of the accident, but these are sort of recreations from what I believe MIT created these images. This is what that injury would actually look like under a CT scan. You can see it's entering just below his left eye and exiting the top of his head. And this little part of the brain that the railroad spike is going through, that's your prefrontal cortex, just totally destroyed in poor Phineas Gage. That being said, even though he had this severe brain trauma, he still lived a somewhat normal life. I mean, his quality of life was worse after this accident, but he was still able to have a basic life. Another great example of brain plasticity would be a hemispherectomy. And I highly encourage you to click on the link here for the hemispherectomy. This is something we talked about in that day one lecture, talking about psychological myths. So a hemispherectomy is when an entire hemisphere, so an entire half of a person's brain, is removed and they still survive. And the video is of a poor young girl who has some debilitating seizures, and the unfortunate way to solve the seizures is to remove the part of the brain that is experiencing the seizures. So they perform a hemispherectomy, and then the video just kind of follows a little bit of her recovery. And it shows that because this procedure was done so early in this child's life, they were able to overcome that trauma and basically live a really normal life because of it, which is just bizarre to think about, the fact that you can live a normal life without half of your brain. All right, so let's move on to the human nervous system. The human nervous system is one way that our brain and our body communicate. In general, we have motor neurons exiting the central nervous system, and we have sensory neurons coming into the central nervous system. All right, we've got this diagram on the right-hand side, and we're going to talk about this at length. But first, we've got the nervous system at the tippy top. You can break that into the peripheral nervous system and the central nervous system. The central nervous system consists of the brain and the spinal cord, which is highlighted in these guys right here. Got the brain, got the spinal cord. Everything outside of the brain and spinal cord is in what we call the peripheral nervous system. So that'd be everything outside of the brain, outside of the nervous system. All these other nerves going to different parts of our body, going to the tips of our fingers, the tips of our toes. You can further divide the peripheral nervous system into the autonomic nervous system and the somatic nervous system. So the somatic nervous system deals with voluntary muscle movements. So if you flex your bicep, that's using your somatic nervous system. If you pick up your foot to step over an obstacle, that involves your somatic nervous system. It controls voluntary muscles. That's in contrast to the autonomic nervous system, which is involuntary internal organs and glands. These are things that we don't have conscious control over. So the autonomic nervous system can be further split up into the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system arouses us, gets us ready for action. The parasympathetic nervous system does the exact opposite. It calms us down. It gets us ready for rest of some kind. All right, so let's talk about these different nervous systems in a little bit more detail. The central nervous system, we've got the brain and the spinal cord. Here's a lovely little diagram that shows a very simple reflex. This object here on the right side of the image represents a cross-section of your spine. And within your spine, you've got those motor neurons exiting, and those motor neurons control your muscles. And you've got sensory neurons entering the spine, entering the central nervous system. And those sensory neurons bring information from your senses. In this case, it would be temperature information from your skin. So let's say you, for whatever reason, feel the desire to touch the flame of a candle. So you reach out for it, and as you get closer, your sensory receptors and your sensory neurons are sending information to the central nervous system saying, hey, whoa, 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 this is hot. This is going to damage our body if we get any closer. So that information travels very quickly to the central nervous system, and we actually have some reflexes that don't need to send information to the brain before action takes place. This is one of those reflexes. So extremely hot temperature information comes in, it's processed very, very quickly in the spine, and the motor neuron sends information to the wrist to pull back very quickly before damage occurs. So great example of sensory information coming in, that information is being acted on, and we avoid damage. Thank you, central nervous system. You're the best. All right, so that's the central nervous system, and that's in contrast to the peripheral nervous system, which is all the nerves outside of our brain, outside of our spinal cord. We have the somatic nervous system within that peripheral umbrella. This consists of voluntary muscle movement. And then we have the autonomic nervous system. This is internal regulation. And we're going to look at sympathetic and parasympathetic nervous system within this umbrella term of autonomic. So the sympathetic nervous system gets the body ready for action. If you ever find yourself in a fight or flight situation, maybe you're walking through the woods at night and you hear a twig break behind you, that is going to cause your sympathetic nervous system to probably act like crazy. Your heart rate's going to increase, your breathing rate is going to increase, you're going to be hyper aware, hyper alert, adrenaline's going to be coursing through your blood, you're going to be ready to run away or fight off some sort of predator. That is your sympathetic nervous system. The parasympathetic nervous system does the exact opposite. It calms us down, gets us ready for bed. Let's say it's the end of the day, we've had a long day, we're ready to go to sleep. Your parasympathetic nervous system is getting activated to sort of calm you down and get you ready to go to sleep. Or let's say you ate a big meal and your belly's feeling really full and you're starting to feel a little bit sleepy. That's your parasympathetic nervous system telling you, hey, let's just chill out for a bit. Let's try to digest all this food that we just put in ourselves. So two different nervous systems that have two very different functions. Sympathetic nervous system gets us ready to do something. Parasympathetic nervous system calms us down. Excuse me. So the parasympathetic nervous system helps with digestion, helps with sleep. It returns our body to homeostasis. Homeostasis is a term that we're going to revisit throughout the course. And homeostasis is this idea of complete balance. You have all of your bodily needs met. You're not hungry, you're not thirsty, you're not in pain, you don't feel the need to go out and do anything because you are perfectly content. Homeostasis is kind of this, I don't want to use the word mythical, but it's sort of an unattainable goal. But it's something that our parasympathetic nervous system is hypothetically constantly striving for. The sympathetic nervous system, again, does the exact opposite. It gets us ready to do something. So because of the sympathetic nervous system, we can increase our heart rate, release adrenaline, increase respiratory rate. Now, these three different actions here are getting the body ready to do something. Increasing your heart rate, you're pumping blood through, you're giving your muscles oxygen and nutrients. The pituitary gland is releasing the hormone of adrenaline into your bloodstream. And with that increased heart rate, it's getting pumped all around your body, getting to your different muscles. Increase your respiratory rate. You're trying to saturate your blood with oxygen so your muscles have all the oxygen they need to run away real fast or punch a bear in the face. So these three different things by the sympathetic nervous system are all mustering resources for your body to do something drastic very fast. So here we have the basic response of the sympathetic nervous system. We have the alarm stage, and this yellow line here represents your arousal level, and this blue line here represents that mythical homeostasis level. So at homeostasis, we're just totally chilling, we're just totally fine, super content. But again, we're walking through the woods, something snaps behind us, and then this yellow line goes all sorts of haywire. We go off this homeostasis line. So in the alarm phase, we are very, very briefly predisposed. But as we get those resources mustered, we start to build up into the resistance stage. This is where we're ready for action. This is where we can push back against that predator or against whatever that stressor is in our environment. But if we stay at this stage for too long, if our heart rate is increased for too long, we have adrenaline in our bloodstream for too long, our respiratory rate is high for too long, we start to experience the exhaustion phase. So let's say we're just hyper aware, super alert, ready for action for 5 minutes, 10 minutes, 15 minutes. We're waiting for that critter to charge us, that predator to run out of the woods and attack us, and we're just ready to pounce back on it. If that happens for too long, we start to feel exhausted. All of these resources that are mobilized to help us act in some way, they start to deplete. And we start to realize, like, hey, if nothing's going to happen here, I got to rest a little bit. I've been at this super hyper alert stage for way too long. Now, the autonomic nervous system evolved to handle temporary stressors. I can't stress that enough. This is a good way for our evolutionary ancestors to deal with the obstacles and stressors in their life. That could be crazy scary predators like woolly mammoths or saber-toothed tigers or other humans. Excuse me. So those stressors are temporary. You encounter them in your environment and you either run away, fight them off, or they eat you. That interaction really doesn't last that long. Nowadays, we don't face a whole lot of stressors like that. We don't face predators chasing us down across the savannah. The stressors that we experience in our life are related to modern living. Things like deadlines, things like upcoming exams, research papers that you have to write for school, trying to land a job or going through a job interview. Those are all modern day stressors, and they are not temporary, they are prolonged. So, in this way, our autonomic nervous system is kind of maladaptive for modern life. It's not helpful to be stressed out and have your sympathetic nervous system activated for days on end before an exam or before a final paper is due or something like that. This system was not built to have sustained levels of stress. It was built to muster up resources very briefly, deal with those stressors, and then continue on our way, hopefully back to homeostasis. So again, the autonomic nervous system was probably great back when the stressors we experienced were temporary. But nowadays, in modern life, our stressors are prolonged. They last for days, weeks, months, bills to pay, things like that. Try to get a good job. Those stressors do not come and go very quickly. All right, so we've talked about the nervous system and the different subcategories. Now, the nervous system is electrical. We've talked about action potentials, excuse me, event-related potentials. The nervous system uses neurotransmitters. Electrical neurotransmitter communication is great for very fast and very brief types of communication. If you need to move your hand in response to a hot candle flame, that needs to be fast, but you also need to be able to move your hand pretty quickly after you've already moved it. So in this example, electrical communication and neurotransmitter communication is pretty ideal. We can move our hand very quickly, and then we can regain control of it pretty quickly after it's moved. Contrast that with the endocrine system. The endocrine system is used for changes that need to occur in our behavior that need to be long lasting. So the endocrine system is basically chemical communication that occurs outside of the nervous system. So I mentioned this earlier in the last lecture, but the difference between neurotransmitters and hormonal is basically nominal. Molecularly, there's not much difference between neurotransmitters and hormones. Hormones occur outside of the central nervous system, neurotransmitters occur within the central nervous system. However, hormones tend to stick around in our bloodstream for a relatively long period of time, which means they have an influence on our behavior the entire time they're in our bloodstream. And it takes a while for the pituitary gland or the liver to filter them out, to suck them back up. Contrast that with neurotransmitters hanging out in a synapse, and they're briefly latched onto a receptor site of a postsynaptic neuron, and then they are reuptake back into the presynaptic neuron. It happens pretty quickly. So the endocrine system is useful for things like that when the sympathetic nervous system is activated and we need to potentially fight off a predator or run away from a predator. We might experience a huge dose of adrenaline in our bloodstream. This will cause us to be alert for minutes on end. That is very useful because that predator might stick around for minutes on end. All right. So the endocrine system consists of glands throughout the body. The pituitary gland is a key one. And these glands secrete hormones into the bloodstream, and those hormones are circulated around the body, and those hormones have different effects on different organs. So in the hypothalamus, we have the pituitary gland. This is sometimes called the master gland. The pituitary gland controls other glands. And some of those other glands would be the adrenal gland. This is in the sympathetic system. This gets us ready for fight or flight. Adrenaline is a very powerful hormone that gets us ready for action. The pancreas is another gland. It regulates blood sugar. This also prepares us for fight or flight. It also kind of regulates our hunger. It tells us, when we should be hungry, when we shouldn't. Kind of gets us hungry when meal times get closer or when we're in the presence of food. Hunger is another one of those behaviors that is good to stick around for a while. You don't want someone to briefly experience hunger and then maybe not eat anything and then go about their day not feeling hunger. Our sex glands, these would be our testes or our ovaries, is another example of the endocrine system. And these release sex hormones. So things like testosterone, things like estrogen. And when they are first activated around puberty in our lives, that's when they start to signal to the body that we should experience the changes of puberty. So that's when secondary sexual characteristics start to develop. And that's when we also start to typically engage in sexual behavior. And this is due in large part to the presence of these hormones that have not existed in our bloodstream in any large number until that point. So, sex, aggression, hunger, hunger related behavior, let's call it, these are all good behaviors to last for a long time, for a relatively long time. Hunger was a great example. Sex is another great example. Aggression is another great example. You don't want those types of motivations to be fleeting. You want them to stick around for a while. You want them to have a long lasting influence on behavior so that the person can go out and run away from that predator or go out and seek a source of food or go out and seek a mate for procreation. Those are all things that are very helpful to have a sustained motivation for. All right, so let's talk just briefly about the difference between hormones and neurotransmitters. As I've said before, the difference is the location. Hormones are located outside of the central nervous system, neurotransmitters are located inside, and the central nervous system is protected by something called the blood-brain barrier. And it's a semi-porous membrane that allows some substances to pass through and others to not. So if you think about the central nervous system as sort of like the command center of the body, it's an incredibly powerful, but a somewhat fragile system. And so this blood-brain barrier is sort of like the filter that allows good, helpful, healthy substances to pass into it and blocks unhealthy, potentially harmful substances. Excuse me. This is actually a pretty big barrier for new pharmaceutical products to overcome. If you are a scientist trying to develop some sort of mental illness drug, one of the first things you have to do is make sure that your drug can be ingested and passed through the blood-brain barrier to actually make it to the brain in the first place. So if a neurotransmitter is on the inside of the blood-brain barrier, we call it a neurotransmitter. If it's on the outside, we typically call it a hormone. Some examples of hormones, we have oxytocin, sometimes called the love hormone. This is pretty involved with feelings of connectedness and affection. Epinephrine and norepinephrine, these are usually under the umbrella term of adrenaline. They stress us out, they get us ready for action, they make us feel hyper aware of our surroundings. Testosterone and estrogen, these would be your sex hormones. They signal the development of body, Jesus, they signal the development of our bodies during that puberty stage. And then when they are present in our bloodstream after puberty, they usually signal sexual motivation. All right, we're going to finish up with just a couple or just one sort of miscellaneous topic here. I want to talk about the two brain hemispheres. We have two of them. We have a left and a right one, and they are connected through the corpus callosum. The corpus callosum is a band of axons. You remember what an axon is from talking about the anatomy of neurons. It's a band of axons that connects the hemispheres. And believe it or not, this is the only place that communication between the hemispheres occurs. It's not like the two hemispheres are just melded together and every single part of one hemisphere is talking to every single part of the other hemisphere. There's just one really thick wire, one really thick band of axons that they talk through. There are cases where it is important to sever the corpus callosum so that the two hemispheres cannot talk to each other. This is usually done to prevent severe epileptic seizures. When this is done, these are often referred to as split brain patients. And I've got another video that I encourage you to click on that will be linked on the Canvas page. This happens in split brain patients when the brain hemispheres can no longer communicate because that corpus callosum is severed. You have some pretty interesting effects because of the loss of that communication. So, for example, you can do these really goofy experiments here. All right, to set this up, we've got the left hemisphere. And the left hemisphere of the brain is sometimes referred to as the language production center. When we produce language, that heavily involves our left motor cortex. That is sometimes referred to as Broca's area. If that is damaged, like it was in our case study of patient Tan, then you will have severe difficulties producing speech. All right, so left hemisphere houses the language production center. So let's say you have information entering the right hemisphere from your left eye, because remember, the hemispheres, for the most part, are contralateral. So if you present something to the left eye, that will go to the right hemisphere. But information can only be vocalized if it's sent to the left hemisphere. So let's walk through that again. Let's say you're able to present information only to someone's left eye. That means it will travel to the right hemisphere. Once it's in the right hemisphere, it has to travel back to the left side of your brain, to the language production center, before you can say what it is that your left eye saw. Now, let's say the corpus callosum does not exist. It's been severed. Your two hemispheres cannot talk to each other. Now, you show information only to someone's left eye. That information enters the left eye, goes into the right hemisphere, and now there's no way for the right hemisphere to get that information over to the left hemisphere where language production can occur. So if that happens, someone can't tell you what they saw in their left eye. But what they can do is point to a sign and tell you what it was if they're given a couple choices. So here we have the basic diagram of this experiment here. So we have the participant come into the laboratory. They look at this screen. They look at the dot right in the center. Briefly, they are flashed a word that is split. So two words separated by a dot are momentarily projected. So the word heart. But on the left side of the screen, we have the word he. On the right side of the screen, we have the word art. And if you are looking directly at this dot in the middle, then your left eye will see the H and the E. Your right eye will see ART. So that happens. That's briefly flashed. And you ask them, what word did you see? More often than not, they will say, I saw the word art. The letters ART appeared on the right side of the screen. Therefore, it entered their right eye, and that information from their right eye was passed on to their left hemisphere, where the language production center is located. So they can produce that information vocally. If you ask them what happened on the left side, what word was presented, they won't be able to tell you. They won't be able to vocalize what it is that their left eye saw because that information is isolated in the right hemisphere. But if you give them the option, if you give them a couple options, you give them a piece of paper with several different choices of what they saw with their left eye, with their right eye, they will be able to point it out. So in this way, we have this very strange circumstance where you can separate the hemispheres, and without that communication, you won't be able to vocalize information that's presented to the left eye. All right. Last thing I want to talk about, and we're going to talk about these more in detail later on, but is brain-computer interface. And once again, this is sort of the frontier of the field. Cochlear implants have been around for a while, but this was sort of the first attempt at brain-computer interface with any sort of widespread success. So for those of you that are unaware, a cochlear implant is a device that allows a deaf person the ability to hear. It is an electrode that is sent into the cochlea, and it's sort of an artificial cochlea that produces the sensation, but not the perception of sound. We'll talk quite a bit about that when we get to the hearing chapter. Ocular implants are starting to become a thing. These are basically bionic eyes. They take in incoming information visually and they send it on to the brain for processing. And then we have robotic limbs. If you click on that link there, which will be linked on the Canvas page, you'll be able to see a monkey in an experiment who's got some electrodes attached to his motor cortex, and those electrodes control a robotic limb. And you can see this cute little monkey go absolutely crazy trying to reach a banana. And there's some pretty cool experiments done where these monkeys are trained in on these robotic limbs. And of course, they go a little crazy and just love to grab those bananas when they're placed within their reach. And obviously, that would be moving on towards human robotic limbs. It's kind of in the testing phase right now, but definitely within our lifetimes, I think we're going to see this a lot more commonly. People that lose limbs for different reasons are definitely going to have the ability to replace them with these sort of robotic limbs. All right, that's all I have for the brain chapter, and thank you for listening.